Hey everybody, and welcome to our video on meiosis. This is part of our Module 2 Part 6 series looking at cell division, cell diversity and cell differentiation. And in this video we'll be having a look at specification reference 2.1.6f, the significance of meiosis in life cycles, and 2.1.6g, the main stages of meiosis. First thing that we need to understand then is why meiosis is actually important in our living organisms. Now, this all ties back in to sexual reproduction. So if we've got organisms that are reproducing through sexual reproduction, then we are going to see increased genetic variation. And the simple reason behind that is that we're going to get recombining of the genetic material from two unrelated members of the same species. And obviously that's something that happens during that process of fertilization. The reason that having increased genetic variation is beneficial to organisms is just that it increases the chance of survival, because when we get some sort of change in the environment, then some individuals are likely to have some kind of survival advantage. It's not that they're undergoing a mutation at the point of the change, it's just that we've got this variation within the population, and therefore some could very well have a better chance of surviving than others. Now, before we delve into the actual processes, we need to understand some key terms here. And there's two that in theory you met back at GCSE, but we're just going to recap to make sure we understand them properly. Diploid is our first one, and hopefully our little biology knowledge tells us that di means two. So diploid just means we've got two sets of those chromosomes, and when we're writing in any kind of diagram or numerical way, we will always use the symbol 2n for that, because we've obviously got 2 times the chromosomes, the n number. Haploid are the ones that have only one set of chromosomes, and therefore we just use the lowercase n to represent any haploid cells. Now, when sexual reproduction is occurring, then the nuclei of two haploid gametes will fuse, and therefore we produce a diploid zygote. So remember, gametes are just the sex cells. The zygote is obviously that fertilized cell. So what we're going from are two haploid gametes to a diploid zygote there. In terms of where meiosis comes into this sexual reproduction process, it's all to do with actually producing those haploid cells. So what actually happens in meiosis is those diploid germ cells in our gonads, so ovaries or testes, depending on obviously your sex, will actually have their chromosome number reduced to half to make those haploid cells. So we're starting off with a diploid cell, so that's our 2n, and we're going to make haploid, so just n. If we put that into the context of humans, then a normal body cell in humans will have 46 chromosomes. Therefore, our diploid germ cells have 46 chromosomes, but those gametes that we're going to be forming are haploid, therefore they will each have 23 chromosomes present. We do have a couple of other terms that we should make ourselves familiar with as well. Homologous chromosomes is the first one. And when we talk about homologous chromosomes, these are just matching chromosomes that contain the same genes at the same loci. However, the actual alleles that are present may vary. And remember, an allele is just a different version of that same gene. So when we're looking at our terms here, obviously alleles we should have met back at GCSE, just meaning the different versions or variants of that same gene. And homologous, again, split it up, where we've got homo, that is same. Okay, so we've basically got this matching going on. Matching chromosomes, and our genes are obviously located at specific points called a locus. Okay, so our many loci will be the positions that those genes are found at on the chromosomes. If we now think about the process of meiosis then, before meiosis can take place, each chromosome has to be duplicated and that takes place in interphase in the S or synthesis phase. Once we've actually replicated all of those chromosomes, then we are going to enter the stages of meiosis 1. Please note, numbers are important when it comes to meiosis. When we did mitosis, there were no numbers. We literally had prophase, metaphase, anaphase, telophase. When it comes to meiosis, we're going to have similar processes going on, but there are going to be 2 stages to meiosis. So number 1, number 2, this is going to be important in any answer we give. So meiosis 1, first bit, prophase 1. Now in prophase 1, we've got our chromatin condensing and chromosomes supercoiling. That nuclear envelope is going to break down. Those spindle threads of tubulin form from the centriole in the animal cells, and we're then going to see these chromosomes coming together in their homologous pairs. And remember, each chromosome is made up of those 2 sister chromatids. One thing that we see happening in our little prophase 1 is a process called crossing over. And crossing over is very important in terms of increasing genetic diversity further, because what we're going to see is an organism's ability to shuffle the alleles that are present on those actual chromosomes. Now, what happens in crossing over is down in this little diagram here. We've got our homologous chromosomes aligned, one's red, one's blue, sitting next to each other. And crossing over, as the name suggests, is where 2 bits of the chromosomes literally cross over with one another. And when they're doing this, what's going to happen is they're actually going to swap part of the chromosome. So what we then form are these recombinant chromatids. So we can see here that we've got predominantly blue on this little arm here, but it then has a little red tip on it. So we've got the same thing with the red, predominantly red, but with a little blue tip. So what we've actually done is we've created these recombinant chromatids. We've actually swapped a bit of the red and the blue chromosomes, therefore we've shuffled the alleles. And remember, we will still have the genes in the same loci, so the same position. So when we're doing this, what we're doing is just changing which alleles for those particular genes are present. We're not changing the genes, we're just changing which alleles for them are going to be there. We then enter metaphase 1, and this is where the pairs of homologous chromosomes attached to the spindle at the equator or the metaphase plate by their centromeres, and this is where we get our next bit of variation occurring in a process we call independent assortment. Now, independent assortment, quite simply, is where those homologous pairs are going to be arranging randomly as to which one faces one side of the cell and which faces the other. It's not a case of, in our diagrams, all of the red ones line up at the top and all of the blue ones at the bottom. That's not life, okay? When we're talking about these homologous chromosomes lining up along that metaphase plate, then it's just completely down to chance as to which one of those chromosome pairs is going to be on the top side of the cell and which one's going to be on the bottom side, for example. So we get this random arrangement of those homologous pairs, which again means that each time this happens it could be unique, and so we get more variation in those potential offspring. Third bit is anaphase 1, and this is where our motor proteins pull the chromosome pairs apart. Centromeres, however, do not divide. Each chromosome consists of 2 chromatids as they are moving apart. What we're going to see here then is a process called allele shuffling, because those crossed over areas are now separating from each other, and so we've got these swapped areas of the chromosome now moving to those opposite poles of the cells. Fourth phase, telophase 1, different in plant cells and animal cells. In animal cells, we get a new nuclear envelope forming around the individual sets of chromosomes now in those poles. The cell will divide by cytokinesis, and we then enter a short interphase as the chromosomes uncoil. If we're talking about plant cells in telophase 1, then quite frequently we're not going to see it. Most plant cells are actually going to jump straight from anaphase 1 to prophase 2. So Animal cells are going to go through this telophase 1, nuclear envelopes forming, cytokinesis, and a short interphase at the end of it. Plant cells typically skip this bit. We're now going to enter stage 2 of meiosis. And what we're going to enter, first bit, is prophase 2. So in prophase 2, the nuclear envelope breaks down, the chromosomes coil and condense, and the spindles form. After prophase 2, we enter metaphase 2. So the chromosomes are then going to be attaching onto those spindles by their centromere at the equator. And again, we've got that random arrangement of the chromosomes. Anaphase 2 comes next, where the centromeres are going to divide. Chromatids are pulled to the opposite poles by those motor proteins. And then into telophase 2, where nuclear envelopes form around those 4 haploid nuclei. Different things happening in animal and plant cells again. Animal cells are going to divide to form 4 haploid cells, but plant cells are going to actually form something we refer to as a tetrad of 4 haploid cells. So when we're talking about a tetrad, it's basically where we've got those 4 cells still kind of joined together. So what we actually see is something that's got its main sort of cell wall around the outside, and then when you look at it, it's kind of like almost a little scope or bullseye thing. And we then got our individual little nuclei in there. So this is our tetrad for our plant cells. So as we said at the beginning of this video, one of the key things about meiosis is it causes genetic variation. And we've obviously mentioned a few little ways in which that happens throughout those individual bits of the process. But let's just recap on them here so that we are clear in how this genetic variation occurs through the process of meiosis. First one, that crossing over occurring in prophase 1, and that's going to lead to shuffling of those alleles. Second way is the independent assortment of chromosomes, and that's anaphase 1. So what we're going to see is a random assortment of the maternal and paternal chromosomes. Third way, independent assortment of the chromatids in anaphase 2. And the last one, once we've made all of these haploid gametes, the process of reproduction requires fertilization, and therefore we've got random fusion. It's completely down to chance as to which of the male cells is going to fuse with which of the female cells. So let's be careful. When you are learning these, let's make sure we have the correct process and the correct part of meiosis, including that number. So crossing over in prophase 1, independent assortment of chromosomes in anaphase 1, independent assortment of chromatids in anaphase 2, and then obviously random fusion of the gametes in the process of sexual reproduction itself. As always, I hope you found this video useful, and don't forget to subscribe to the channel so you can see when the next video is uploaded. And of course, head on over to the website where you can find a range of other resources designed to help you in your A-level biology studies.